Hi, my name is Peter Schulam, and I'm an author of Disease Trajectory Maps, a paper appearing at NIPS 2016. Many complex diseases are driven by a poorly understood combination of genetics, environment, and behavior, which makes them challenging to treat. Our work aims to help medical scientists better understand these complex diseases by discovering low-dimensional structure in clinical time series data. As a motivating example, we studied the systemic autoimmune disease scleroderma. Scleroderma is named for its effect on the skin, but the underlying mechanism can cause serious damage to many internal organs as well. However, which organs are involved and how they are affected varies from person to person, and there are no known biomarkers that can predict behavior. Instead, clinicians use laboratory tests recorded over time to monitor the disease and to classify individuals into subtypes, which are subpopulations that express the disease similarly. There are two widely accepted subtypes in scleroderma that are defined by the trajectory of skin disease over time, but there is still tremendous variability within these two subpopulations. Instead of clustering individuals, our work aims to discover structure in the heterogeneous population by learning low-dimensional representations of disease trajectories. More specifically, we embed sparse and irregularly sampled clinical time series data into a Euclidean space where proximity implies that the two individuals express the disease similarly. Our approach builds upon the linear mixed model, a widely used model for clinical trajectory data. Here we show some sample trajectories of scleroderma-related lung disease. Each of the 9 grid cells represents the lung function for one individual over a period of 12 years. Each dot is a single laboratory test result. Higher values indicate better lung function. Linear mixed models assume that trajectories lie within the span of a finite dimensional function space, and jointly learn the coefficients of all individuals in that space. One can show that linear mixed models are learning a linear function from a low-dimensional space to the coefficients. We build upon this insight and allow the function to be nonlinear using the kernel trick. Disease trajectory maps allow us to organize large collections of raw disease trajectory data, like that mess on the left, by projecting into a low-dimensional space that is easy to visualize, demonstrated in the scatter plot on the right. By plotting trajectories of groups of individuals with nearby representations, we are able to uncover nuanced variations of the two widely accepted scleroderma skin disease subtypes. Moreover, we discover surprising trajectory shapes that are not well explained by the existing subtypes. In the paper, we further discuss our qualitative findings for both skin and lung disease, and quantitatively analyze the relationship between our learned representations and important clinical outcomes. Thank you for listening, and please come see us at our poster in Barcelona.